In humans, biological sex is determined when a sperm penetrates an ovum. However, a honeybee operates on a different logic. A solitary, unfertilized egg can bypass the father entirely, dividing and developing directly into a male. This reveals that sex determination is an emergent, mechanical outcome of chromosomal sorting rather than a universal biological law. For centuries, the mechanism remained a philosophical mystery. Understanding only began once cytologists tracked physical structures inside dividing cells. In 1891, H. Henking observed a specific, distinct nuclear structure that persisted throughout insect spermatogenesis. Henking labeled this the X-body. He observed that exactly 50% of the resulting sperm cells received this structure, while the other half did not. Later investigations confirmed this X-body was a chromosome. Its discovery proved that sex could be measured and predicted through physical cellular architecture. To understand these variations, we must map the 4 distinct genetic architectures found across nature. These systems form a comparative framework: XO, XY, ZW, and haplodiploid. Most chromosomes form identical pairs called autosomes. The specific pair dictating sex is categorized separately. This relies on heterogamety, a biological asymmetry where one parent produces 2 different gametes. By evaluating each quadrant against these variables, we can mathematically prove which parent dictates the future of the offspring. In the XO system of the grasshopper, the female possesses a matched pair of X chromosomes. The male, however, is missing the second chromosome. He carries only a solitary X. During spermatogenesis, this single X chromosome cannot split evenly among all resulting cells. This produces 2 types of sperm. 50% carry the X chromosome, while 50% carry no sex chromosome at all. This chromosomal inequality is defined as male heterogamety. Eggs receiving the X-bearing sperm become females, while those fertilized by the empty sperm become males. Under the XO architecture, maleness is dictated by a definitive physical absence of genetic material. The second quadrant covers the XY system used by mammals and fruit flies. In this system, researchers identify the sex-determining pair by mapping the total chromosomal count. Unlike the XO model, human males and females share the same total number of chromosomes, with 22 identical pairs of autosomes. The variation exists in the sex chromosomes. Females have a matching XX pair, while the male counterpart is a distinctly smaller Y chromosome. As a homogametic parent, the human female produces only one type of egg, each carrying an X chromosome. Human males demonstrate heterogamety, producing a 50-50 split of X-bearing and Y-bearing sperm. Because the maternal contribution is constant, the genetic payload of the father sperm acts as the absolute determinant of sex. In the avian ZW system, however, the primary mechanical authority definitively shifts from the sperm cell to the egg. While birds also maintain equal chromosome counts between sexes, cytologists use the letters Z and W to designate their specific architecture. In this model, the male is the homogametic parent, possessing a symmetrical ZZ pair and producing identical sperm. The female possesses the unmatched ZW pair. This creates female heterogamety. The probability of the offspring's sex is decided within the ovum rather than during spermatogenesis. Within the ZW architecture, the genetic makeup of the egg exercises absolute control over the outcome. The final quadrant removes the requirement for specialized sex chromosomes entirely. In the haploid-diploid system of the honeybee, sex is determined strictly by ploidy, the total number of chromosome sets received. A fertilized egg containing 32 chromosomes develops into a diploid female. An unfertilized egg develops through parthenogenesis into a haploid male containing only 16 chromosomes. Because the male is already haploid, he produces sperm through mitosis. Cloning is 16 chromosomes without the segregation seen in meiosis. This creates a unique pedigree where a male bee has no father and can have no sons. His lineage links only to grandfathers and grandsons through his mother and his own female offspring. Haplodiploidy proves that sex can be dictated by the raw presence or absence of fertilization itself. Viewing all 4 systems simultaneously reveals a consistent biological pattern. In the XO and XY systems, the outcome is isolated to the sperm. In the ZW system, the variable is held by the egg. In the haplodiploid system, the decision rests on whether fertilization occurs at all. Across all architectures, sex is dictated by a mechanical asymmetry in genetic contribution. Isolating the human application clarifies the biological reality of our own reproduction. Male spermatogenesis produces an unyielding 50-50 distribution of X and Y chromosomes. This data directly contradicts the historical tendency to blame or ostracize mothers for their children's sex. Cytological evidence clarifies that mothers provide only a constant X chromosome. The genetic makeup of the father's sperm is the only variable that determines the outcome. The empirical data from cellular observation provides the weight required to dismantle false societal narratives.